Hello everyone, myself Abhijit Sashmal. I am from Department of Bioinformatics of Maulana Abul Kalam Azad University of Technology. Today I am going to discussing about bioinformatics tool Cytoscape. So now I will show you my PPT. So here the topic is Cytoscape. Now this is the index. First is introduction. So Cytoscape is a open source bioinformatics software platform from visualizing molecular interaction networks and integrating with gene expression profiles and other state data. Cytoscape is also has a JavaScript centric sister project named Cytoscape.js that can be used to analyze and visualize graphs in JavaScript environments like a browser. So firstly, we have to install Cytoscape tool. Now we have to go a browser like Chrome, you can use Brave or etc. etc. Now search Cytoscape, download. Okay. So now this is the first link you have to open. Then this icon for download 64-bit. If you are a Windows user, then you have to click it and download it. So I already download the Cytoscape tool. So this is my home page of Cytoscape tool. Okay. So Cytoscape, it is used for gene-gene interaction, protein-protein interaction, and also gene-protein interaction, and so on. So first we need to, if you want to interact the protein-protein or gene-gene, then you have to create a file in Excel. So this is my Excel sheet. This is my Excel sheet. You can see. Then we have to go the file option, import network from file. Now this is my protein. So this is our target. These are protein names. Target proteins. It's are okay. Yes. So this is the interactions. You can scroll it out, zoom out, zoom in, zoom out. You can do this. So this is this Cytoscape working like this. Now we have to go our PPT. This is our introduction part. Now the history. Cytoscape was originally created at the Institute of System Biology in Seattle in 2002. Now it is developed by an international consortium of open source developers. Cytoscape was initially made public in July 2022. The version is 0.8. The second release in version 0.9 was in November 2002 and version 1.0 was released in March 2003. Version 1.1.1 is the last stable release for the 1.0 series. Version 2.0 was initially released in 2004. Cytoscape 2.83. The final 2.xx version was released in May 2012. Version 3.0 was released in February 1st, 2013, and the latest version 3.0.32 was released in August 2013. Now going to next slide. Gene plugin. What is gene plugin? The Gene Mania Cytoscape plugin brings fast gene function prediction capabilities to the desktop. Gene Mania identifies the most related genes to a query gene set using a guild by association approach. The plugin uses a large databases of function functional interaction network from multiple organisms, and each related gene is traceable to the source network used to make a prediction. User may add their own interaction networks and expression profile data to complement or override the default data. The development, the Cytoscape code developer team continues to work on this project, and in the near future is going to release next major version, Cytoscape 3.0. It will be a more modularized, expandable and maintainable version of Cytoscape. The uses of Cytoscape, while Cytoscape is most commonly used for biological research application, it is agnostic in terms of uses. Cytoscape can be used to visualize and analyze network graphs of any kind involving nodes and edges, etc. social networks. A key aspect of the software architecture of Cytoscape is the use of plugins for specialized features. Plugins and developed by core developer and the greater user community. Going to next slide. Features. Load the save previous constructed interaction networks in GML format. Load and save networks and node attributes views in XML document format called XGMML. Input mRNA expression profile from tab or space delimited text files. Load and save attributes on nodes and edges. For example, input a set of custom annotation uh, terms of your proteins, create a set of confidence values for your protein-protein interaction. Import gene functional annotation from the gene ontology and cake databases. Cake database is a pathway database. It is used for molecular docking. The next point is directly import geoterms and annotations from OBO and gene association files. Going to next slide. It is analysis. Now this is the analysis. Plugins variable for network and molecular profile analysis. For example, filter the network to select subset of nodes based on the current data. For instance, user may select nodes involving in a threshold number of interactions, nodes that share a particular geo annotation, or nodes whose gene expression level change significantly in one or more conditions according to p-values loaded with the gene expression data. Going to next slide. Find activate sub networks or modules. The network is screened against gene expression data to identify connected sets of interactions. That is interaction sub networks whose genes show particularly high levels of differential expression. The interaction contained in each sub network provide hypothesis for the regulatory and signaling interactions in control of the observed expression changes. Now this is the conclusion. Cytoscape is an important software to visualize graphs in JavaScript like a browser and molecular interaction. It also has an importance in the uses of biological research sciences. In short, it helps in the analysis of node and edges in the networks. So, okay, this is my Cytoscape tutorial. I think you will know too much from this video. So, okay, goodbye.